Biology 101, Lecture 12. Hello, students. Today we're going to look at the molecular basis of inheritance. So it's no longer Mendel's laws. We're going to look at the really deep down. We're going to dig in there and find out what actually happens. Accuracy in copying DNA is pretty amazing. One mistake is made in about 10 raised to 10. That is more than a billion. Very, very high fidelity. Balance between complete fidelity and a low mutation rate has been, over long periods of time, allowed the evolution of the rich diversity of species that we see today. So it isn't perfect. There is one mistake made every so often, and it's slow enough and little enough that the mutation is done over a very long period of time, but it allows evolution. Synthesis of new strand of DNA is in the replication bubbles. The direction is 5' to 3' with a leading and lagging strand. And we're going to look at that. And we're also going to look from DNA to proteins. Transcription and translation, what is it? How do we go into those details? And the central dogma, which is DNA to RNA to proteins. That is the central dogma. Replication bubbles, what are they? Well, when DNA has to, a gene has to be expressed or DNA has to duplicate, the enzyme RNA polymerase pries apart two strands of DNA, attaches to DNA at the promoter site, and joins together RNA nucleotides complementary to the template strand. Replication begins at sites called origins of replication, where the two DNA strands are separating, opening up a replication bubble. There may be hundreds or even thousands of origins of replication on one chromosome when this is happening. Replication begins in both directions from each origin until the entire molecule is copied. And here's a picture of exactly that. So on the left-hand side, we're replicating an E. coli DNA. And you can see how there's a bubble, a replication fork, which just grows into two. On the right-hand side, there's a replication in a eukaryotic cell. And there's this very small circle drawn, which is the origin of replication. And every origin of replication is going to have this bubble and a replication fork, and you end up with two daughter DNA molecules. At the end of each replication bubble is a replication fork, a Y-shaped region where new DNA strands are elongating. Enzymes called helicases untwist the double helix at the replication forks. Single-strand binding proteins bind to and stabilize single-stranded DNA, because DNA likes to be in a double strand, and it would be very unstable if it just sat around for a while as a single strand. And topoisomerase corrects overwinding ahead of replication forks by breaking, swiveling, and rejoining DNA strands. So it's a magical enzyme that does this for us. And here we have topoisomerase, and it is going and fixing the DNA, but we have primase, which is working its way towards the replication fork, and single-strand binding proteins stabilizing the opening of the DNA double helix. The rate of elongation is pretty fast. It goes at a pretty fast clip. It's about 500 nucleotides per second in bacteria, but only about 50 per second in human cells. Each nucleotide that is added to a growing DNA strand is a nucleoside triphosphate. DNA polymerases add nucleotides only to the free 3' end of a growing strand. Therefore, a new DNA strand can elongate only in the 5' to 3' direction. And here's a picture of that, a leading strand and a lagging strand. These are two strands of DNA, and each one has a leading and a lagging strand and the origin of replication and the overall directions of replication. DNA polymerase is starting to synthesize a leading strand here. There's a parental DNA on the left-hand side, and it's just like unzipping a zipper. So it just continuously elongates in the 5 to 3' direction. Along the template strand of DNA, DNA polymerase synthesizes a leading strand continuously, moving towards the replication fork. To elongate the other new strand, called the lagging strand, DNA polymerase must work in the direction away from the replication fork. The lagging strand is synthesized as a series of segments called Okazaki fragments, which are joined together by DNA ligase. So this is not quite the same way as the leading strand works. Here is a summary of what we just talked about. Helicase, the enzyme that unwinds the double helix at replication forks. Single-strand binding proteins, which stabilize the single-stranded DNA. Topoisomerase, which prevents overwinding ahead of the forks, and it breaks and swivels and rejoins DNA strands. And primase, which synthesizes an RNA primer at the 5' end of a leading strand and at the 5' end of each Okazaki fragment of the lagging strand. Continuing on with the summary of we just talked about, DNA polymerase 3, it uses the parental DNA as a template and synthesizes new DNA by adding nucleotides to an RNA primer. DNA polymerase 1 removes RNA nucleotides of primer from the 5' end and replaces them with DNA nucleotides. And DNA ligase joins up the Okazaki fragments of the lagging strand, and on the leading strand joins the 3' end of DNA that replaces primer to the rest of the leading strand DNA. What really happens is that there's a lot of proofreading, replacing, and repairing going on when new DNA or RNA is being synthesized. DNA polymerases proofread newly made DNA, replacing any incorrect nucleotides. Kind of cool, no? This is very smart. In a mismatch repair of DNA, repair enzymes correct errors in base pairing. In nucleotide excision repair, a nuclease, which is another enzyme, comes around, cuts out, and replaces the damaged stretches of DNA. So we've got it all covered. Telomeres, these are really cool things. The usual replication machinery provides no way to complete the 5' ends. It's just not done. So repeated rounds of replication produce shorter DNA molecules with uneven ends. This is not good. Eukaryotic chromosomal DNA molecules have special nucleotide sequences at their ends called telomeres. Telomeres do not prevent the shortening of DNA molecules, but they do postpone the erosion of genes near the ends of the DNA molecules. In a bacterium, the DNA is supercoiled and found in the nucleoid because it's a prokaryote, so there's no nuclear membrane. It's just sort of dumped in one side of the cell. In the eukaryotic cell, DNA is precisely combined with proteins in a complex called chromatin. Chromosomes fit into the nucleus through an elaborate multi-level system of packing. At interphase, chromatin is organized mostly into a 30 nanometer fiber. Some is organized in a 10 nanometer fiber, though folding and looping, but it doesn't get entangled. That's the magic part of it. And this is what it looks like. This is when chromatin is packed in a eukaryotic chromosome. So notice from the left-hand side, the DNA, blue, double helix, and all these proteins, histones, come and attach to it. And then they wrap around this double helix and it looks like beads on a string. So this is a 10 nanometer fiber. But then that fiber wraps over itself and it becomes a 30 nanometer fiber. And then you get even more loops and scaffold. That's a 300 nanometer fiber. And ultimately, you get the entire chromosome, which is about a 1400 nanometer fiber. It's pretty thick. at metaphase. So if we look at it again, these are the histones wrapping that DNA around itself, and then that beaded look wrapping itself over and over. So you get this big 30 nanometer fiber. And then that fiber itself looping and creating a scaffold to become a 300 nanometer fiber, which then folds some more and becomes a 1400 nanometer fiber. So from the DNA template strand, we have transcription, and with that we get a messenger RNA molecule, which then goes and translates the DNA message into a protein. And we're going to look at that next. Transcription. So how do we initiate it? There are 3 stages of transcription: initiation, elongation, and termination. 2 strands of DNA, identical but run anti-parallel to each other. Only one is used for transcription, and that is called the template strand. A promoter of a gene has a transcription start box called a start point called a TATA box. RNA polymerase binds in a precise location and orientation on the promoter, which determines where transcription starts and which of the 2 strands acts as the template. The flow of information from gene to protein is based on a triplet code, a series of non-overlapping 3 nucleotide words. Messenger RNA nucleotide triplet is also known as a codon. There's a triplet code for each of the 20 amino acids. Messenger RNA is complementary and is synthesized in the anti-parallel direction, which is 5' to 3'. There are 64 triplets. 61 code for actual amino acids. 3 triplets are stop signals to end translation. There's always a start codon, which is always AUG, which codes for methionine. And there are 3 stop codons. There is redundancy in the genetic code, but there's no ambiguity. No codon specifies more than 1 amino acid. Codons must be read in the correct groupings or reading frame in order for the specific polypeptide to be produced. Well, that's very logical because with all those letters, if you start from one, you end up with a different word. But if you start like 2 down, you will end up with maybe a different word or nonsense. So the correct reading frame is very important. Here is a codon table for messenger RNA. And so you can see the start codon in green and the 3 stop codons in red. And then all the codons that are coding for the various amino acids. This is how it starts. And there's initiation, elongation, and termination. And you can see that the transcription unit, the promoter will start at the start point, RNA polymerase will act on it, and the whole transcription unit is to follow. The DNA starts, RNA polymerase will go and unwind this. RNA will have a transcript, and the DNA will then keep on rewinding, and you will end up in the direction of the transcription, which is downstream, and you will get a completed RNA transcript, which is the orange strand that you see getting longer and longer. As RNA polymerase moves along the DNA, it untwists the double helix 10 to 20 bases at a time and starts transcribing at the start point. As RNA polymerase moves along the DNA, it untwists the double helix, exposing 10 to 20 DNA nucleotides at a time. But this is duplicated. Oh well. Nucleotides are added to the 3' end of the growing RNA molecule. New RNA molecule peels away from its DNA template, and the DNA double helix reforms. The transcription rate is about 40 nucleotides per second in eukaryotes. Here is transcription, the part called elongation, where we're actually elongating the new strand, the newly made RNA. To make protein in large amounts, the gene is simply transcribed several times by simultaneous RNA polymerase molecules following each other like trucks on a convoy. So you just transcribe the gene several times. Termination of transcription. In bacteria, the polymerase stops transcription at the end of the terminator, and the messenger RNA can be translated without further modification. So that's pretty simple. But in eukaryotes, it's not so simple. RNA polymerase II transcribes the polyadenylation signal sequence. Then the RNA transcript is released 10 to 35 nucleotides past this polyadenylation sequence. So there's an extra part that's transcribed which isn't needed. So then messenger RNA is processed before sending out. Both ends are modified. A cap is added at the 5' end, and a tail is added to the 3' end. Not only that, RNA will have protein coding regions interspersed with UTRs, which are untranslated regions, which have other functions, like ribosome binding. Long, non-coding stretches of nucleotides between coding regions are called intervening sequences, or introns, and those are spliced out. They're just cut out. They're excised. Regions that are expressed are called exons. So introns will allow exon shuffling and increase the probability of crossing over, but what you end up with is a strand of all coding RNA. RNA splicing removes introns and joins exons, creating an mRNA molecule with a continuous coding sequence. Splicing is done in a complex called a spliceosome. A single gene can encode more than one polypeptide depending on which segments are treated as exons. Alternative RNA splicing, so more proteins can be produced per gene. Here is messenger RNA processing. You can see that both ends are processed. A cap is added to the 5' side, and a tail is added to the 3' side. But notice a whole bunch of stuff is cut out. The faded orange are introns, and those pieces are just snipped on out. And then the coding regions, which are the exons, are joined up together, and you have a much smaller version of the gene transcribed, but it's all coding. This is just a picture of a spliceosome, and the pre-messenger RNA being spliced inside it for stability. Okay, so that's just what goes on in there. Translation. In prokaryotes, translation of messenger RNA can begin before transcription has finished. But in eukaryotes, the nuclear envelope separates transcription from translation. Reading the genetic message is done not by messenger RNA, but by transfer RNA, which is a translator. tRNA transfers amino acids from the cytoplasm to the polypeptide chain in the ribosome. The cell always has a stock of all 20 amino acids, so it just pulls them in. A tRNA molecule consists of a single RNA strand, about 80 nucleotides long, and it looks sort of like a clover leaf if you put it in, if you squash it down. Each tRNA molecule translates a mRNA codon to an amino acid. So tRNA has a nucleotide triplet on one end, which pairs with the codon on the messenger RNA, and an amino acid on the other end. It reads a nucleic word and it translates it into a protein word. Kind of cool, no? So here we are. If you look at the two-dimensional structure on the left, which looks like a clover leaf, you will actually see the amino acid attaching site on the very top and the anticodon on the bottom, which is where it binds to the RNA. So on the very right-hand side, it's drawn as a cartoon, so it's easier to see the amino acid site and the anticodon site. tRNA just moves to mRNA where codon by codon amino acids are added on. Enzymes help, and pairing of transfer RNA anticodon with the messenger RNA codon means that there should be one type of tRNA for each mRNA codon for an amino acid. But instead of 61, there only may be 45 tRNAs because some tRNAs are able to bind to more than one codon. And this is cool because this is known as the wobble effect. So this is because the third nucleotide base of a codon and the corresponding base of the transfer RNA can pair with either base, and this is called wobble. So wobble allows redundancy in the genetic code, but not ambiguity. Ribosomes help transfer RNA anticodons bind to messenger RNA codons for protein synthesis. There are two ribosomal units, large and small. They're made out of proteins, so about 1/3 of the ribosome is made out of proteins, and the ribosomal RNA, but the units attach only when a strand of messenger RNA comes along. Ribosomal RNA is the most abundant type of cellular RNA. Ribosomes have a binding site for messenger RNA and 3 binding sites for tRNA. The binding sites are called A, P, and E. A ribosome has 3 binding sites for tRNA. The P site holds the transfer RNA that carries the growing polypeptide chain. The A site holds the transfer RNA that carries the next amino acid to be added to the chain. And the E site is the exit site where the discharged transfer RNAs leave the ribosome. And here is a picture of that. So on the very top, you can actually see a computer model of a functioning ribosome. Ribosomes have 2 parts, notice, a small subunit and a large subunit. So the blue one is the large subunit, and then there's a small subunit at the bottom. And the brightly colored EPA sites are the tRNA molecules. And you see a strand of a messenger RNA going through those tRNAs in this ribosome at the bottom, and at the top, through the exit tunnel, there's a polypeptide like beads on a string exiting. And you see the same thing on the bottom left-hand side, but it's drawn in a cartoon version. Ribosomes start translating at the start codon to start the reading frame. Amino acids are added in only one direction until the stop codon is reached. This causes, once it reads the stop codon, it causes the addition of a water molecule, which hydrolyzes the polypeptide at the P site and releases it. And the water is plentiful. It's always in the cell, so it's easy to go drag one molecule of water in there. Polypeptide will then fold with the help of a chaperone protein, and post-translational modifications occur, like adding sugars or lipids or removing amino acids or cleaving it in half, etc., etc. So there's some processing that has to happen after that ribbon has left the ribosome. Free ribosomes synthesize proteins in cytoplasm. Bound ribosomes make endomembrane proteins, but ribosomes can move around in the cell and change form from being free to bound. A polypeptide chain growing from a ribosome can cue the ribosome to attach to the endoplasmic reticulum if it has a signal peptide. There are Like transcription, there are 3 translation stages too: initiation, elongation, and termination. Initiation brings together the messenger RNA, a tRNA with the first amino acid, and the 2 ribosomal subunits. Then the small subunit moves along the messenger RNA until it reaches the start codon, which is always AUG. And we know that that codes for methionine. And here we are. Translation is occurring. So the small subunit is binding to the messenger RNA on the left-hand side, and the large subunit completes the initiation complex, and you have this configuration. During elongation, amino acids are added one by one to the C terminus. Each addition occurs in 3 steps: codon recognition, peptide bond formation, and translocation. Energy expenditure occurs in the first and third steps. Translation proceeds along the messenger RNA in the 5' to 3' direction. And here is a picture of the whole process. So number 1, there's codon recognition. Number 2, there's a peptide bond formation. Number 3, there's translocation, and then finally the ribosome is ready for the next tRNA molecule. So here it is in another way. This way is probably more intuitive. So a ribosome reaches a stop codon on the messenger RNA, and here it is going to have a release factor which promotes hydrolysis, and the whole thing is going to fall apart. So the protein has been translated, and the ribosomal units and everything else just falls apart, and the polypeptide just floats away. And that is another way of looking at it. So this is a ribosome that's fully functioning, and it is creating a protein. As you can see, it's adding on amino acids, and the message is being read. Multiple ribosomes can translate a single messenger RNA simultaneously, and here we are. They're all clung to one messenger RNA strand, and they're making multiple strands of proteins. So because only one ribosome can make only one protein. So if you want 6 copies of that protein, then 6 of them are going to attach. So this is transcription and translation in a nutshell. It just shows you how it works. Mutations. Point mutations are small scale. It's just one or a few base pair. Could just be a 1 base pair. Single nucleotide substitutions usually have no effect and are silent mutations, or can change one amino acid to another and are called missense mutations. If a single nucleotide change results in an amino acid changing to a stop codon, then that is a problem, and it is called a nonsense mutation, because suddenly the protein won't code, and all of a sudden you have a little bit here and nothing else. So that's a nonsense mutation. So these are point mutations, and they do happen. We do have sickle cell anemia, and that is a point mutation, just one base. We can also have substitutions. A nucleotide pair substitution replaces one nucleotide and its partner with another pair of nucleotides. Silent mutations will have no effect on the amino acid produced by a codon because of redundancy in the genetic code. Missense mutations still code for an amino acid, but not the correct amino acid, which is a bummer. Nonsense mutations change an amino acid codon into a stop codon, nearly always leading to a non-functional protein. So that's always bad. Mutations can be nucleotide pair insertions or deletions involving one or more pairs. These alter the reading frames, and they're called frame shift mutations. This results in extensive missense, usually ending in nonsense and ending with a non-functional protein. So a frame shift mutation is a really bad one. So this is a missense mutation. So missense mutation, here we are. Instead of one base, there's another base, so it's different. And here we have all of them. So we have a nucleotide pair substitution on the left, silent, missense, and nonsense. Or you can have a nucleotide pair insertion or deletion. You could have a frame shift mutation where one nucleotide pair is inserted, or you could have a frame shift mutation where one nucleotide pair is deleted, or you could have a 3 nucleotide pair deletion, and so that's just going to make it complete nonsense. So we've been talking about genes and alleles all this time. It's probably high time we actually defined it. A gene is a discrete unit of inheritance. A gene can be defined as a region of DNA that can be expressed to produce a final functional product that is either a polypeptide or an RNA molecule. Differential expression of genes. Different cells express different genes according to their function. DNA that is not expressed is inactivated by adding methyl groups. The rate of gene expression can be strongly increased or decreased by binding of specific transcription factors. Some control elements are close, others are far away in the gene. Some repressors recruit proteins that remove acetyl groups from histones, reducing transcription or even silencing the gene. Co-expressed genes, such as genes coding for enzymes of a metabolic pathway, are scattered over different chromosomes. So gene expression depends on a specific combination of control elements. Genes with the same sets of control elements are activated by the same chemical signals. This explains why the number of genes in humans, which is the same number as a mustard plant or a nematode or even a sea anemone, so we don't have a lot of genes, so if you're going by the gene number, we would be just as stupid as plant. But this is why, because there are different control elements which are activated by chemical signals, that the genes in humans can produce so many different proteins. Messenger RNA degrades in a few minutes in bacteria, but takes hours, days, or weeks in eukaryotes. Proteins tagged for destruction have ubiquitin attached to them. So that's a nice name. And any protein that is doomed will have this ubiquitin attached to it. Only 1.5% of the total DNA of any organism codes for proteins. Isn't that a staggering number? That is an amazing number. So what does the rest do? We don't know yet. After cell determination has occurred, a cell will differentiate into its determined fate and will only express those proteins suited for its determined function. A cell can regulate the production of enzymes by feedback inhibition or by gene regulation. One mechanism for control of gene expression is the Operon model. A cluster of functionally related genes can be coordinately controlled by a single on-off switch. And this switch is actually just a segment of DNA called an operator, usually positioned within the promoter. Here we are. Here's a picture of regulation of a metabolic pathway and an operon. Really nice concept. An operon is the entire stretch of DNA that includes the operator, the promoter, and the genes that they control. The operon can be switched off by a protein repressor. Pretty powerful, yes? The repressor prevents gene transcription by binding to the operator and blocking RNA polymerase. Not good. The repressor is the product of a separate regulatory gene. So here we are, and this is the entire thing is called an operon. It includes the promoter, it includes the gene, and it includes the operator. The repressor can be in an inactive or active form depending on the presence of other molecules. A co-repressor is a molecule that cooperates with a repressor protein to switch an operon off. A repressible operon is one that is usually on. Binding of a repressor to the operator shuts off transcription. An inducible operon is one that is usually off. A molecule called an inducer inactivates the repressor and turns on transcription. Here we are. This is a repressible operon. So genes expressed, genes not expressed. And here is an inducible operon, and here are the genes expressed. And here's a very specific operon. It's a tryptophan operon. And we're seeing if there's no tryptophan, what happens. If there is tryptophan and the repressor is active, what happens? There is another operon which is pretty famous. It's called the lac operon. It's an inducible operon and contains genes that code for enzymes used in the hydrolysis and metabolism of lactose. By itself, the lac repressor is active and switches the lac operon off. A molecule called an inducer inactivates the repressor to turn the lac operon on. And here's a picture of the lac operon operating. Inducible enzymes usually function in catabolic pathways. Their synthesis is induced by a chemical signal. Repressible enzymes usually function in anabolic pathways. Their synthesis is repressed by high levels of the end product. Regulation of the trip and lac operons involves negative control of genes because operons are switched off by the active form of the repressor. Genes within highly packed heterochromatin are usually not expressed because they're hard to get to. Chemical modifications to histones and DNA of chromatin influence both chromatin structure and gene expression. Addition of histones and phosphates loosens chromatin, allowing transcription, while addition of methyl groups condenses it. So here we are, histone acetylation, and that promotes loose chromatin, which permits transcription. Loops of chromatin extend from individual chromosome territories into specific sites in the nucleus. Loops from different chromosomes may congregate at particular sites, some of which are rich in transcription factors and RNA polymerases. These may be areas specialized for a common function. Transcription alone does not account for gene expression. Regulatory mechanisms can operate at various stages after transcription. Such mechanisms allow a cell to fine-tune gene expression rapidly in response to environmental changes. And that is the end of this lecture. See you in the next lecture, students.